and movement. What is it? Locomotion and movement. Now, the movement is the significant feature of the any living organism. What is the first significant property of the living organism? Movement is the significant character of the living organisms. Animals and plants exhibit wide range of movement. Now the plants only fixed body, they are the stationary bodies. They are only fixed in the one place. But in the case of animals, they can move from the one place to the other place. Okay, now coming to the first one, the streaming of protoplasma in the unicellular organism is nothing but amoeba. What is it? Amoeba. Now you can take amoeba. It is a no shape. Amoeba means none. It does not have any shape. The protoplasma, the inside the cytoplasma, it is modified into the pseudopodium. What is that? Pseudopodium. This pseudopodium act like a false feet. Now pseudopodium, it is act like a false feet. With the help of pseudopodium, the amoeba move one place to the other place. Now, for example, it is the amoeba cell or unicellular cell. Inside the protoplasma, modified into the pseudopodium, they move from the one place to the other place. One place to the other place. Coming to the simple form of that is a simple form of the movement in the amoeba. Movement of cilia and flagella, tentacles shown in the many organisms. Cilia. And the cilia movement is takes place in the paramecium. What is it? Cilia means paramecium. Paramecium. Now the paramecium outer surface body consists of the entire outer surfaces. The small hair-like structures is known as cilia. With the help of cilia, they can move from the one place to the other place. Coming to the flagella. Now this flagella present in the euglena. Now the cilia movement, it is present in the paramecium. Next one, flagella. What is it? Flagella. This flagella movement takes place in the euglena. What is it? Euglena. This flagella movement takes place in the euglena. The next one, tentacles. The tentacles are located in the tentacles located in the hydra. Tentacles hydra. These are the cnidarians organisms. They consist of the tentacles. With the help of tentacles, they move from the one place to the other place. Now the paramecium move one place, other place. Cilia. The euglena move one place, other place. Flagella. Now tentacles. With the help of tentacles, the hydra move one place to the other place. Okay, now coming to the human beings can move from the limbs, jaws, eyelids, tongue. Some of the movements result in the changing of the place or location. What is it? Changing the place or location. Such voluntary movement. Movements means voluntary means move one place to the other place. Okay, voluntary movements, that is called as a locomotion. Locomotion means the body, changing the body position. Location is known as a locomotion. Walking, running, climbing, flying, swimming, all forms of the locomotory movements. These all are the locomotor movements also. Locomotory movements. These are the locomotory movements. Locomotory movements. The locomotory structures need not be different those affecting the other types of movement. For example, paramecium, cilia helps in the movement of the food capture as well as locomotion. This cilia, it is helps in the paramecium, not only capturing the food material, they move from the one place to the other place. One place to the other place. Now this cytopharynx, it is known as the mouth of the paramecium is known as a cytopharynx. Now the mouth of the paramecium is known as a, the air-like structures is known as a outer surface of the paramecium is consists of the air-like structure is known as a cilia. What is known? Cilia. Okay, inside the cytopharynx. What is this known? Cytopharynx. This cytopharynx is also known as a mouth of the paramecium. Mouth of the paramecium. Now the mouth of the paramecium, it is only capturing the food material, and also this cilia, it is helpful for the movement. This cilia helpful for the movement. Okay, now next one. Hydra can be used tentacles for the capturing of the prey and also movement of the locomotion. Now hydra, we can indication of the hydra. The tentacles. What is these tentacles? These tentacles are helpful for the capturing the food material and also helpful for the locomotion. And also, see now, you use limbs for the changing the body posture as well as locomotion. What is the use of the limbs? They move forelimbs and hind limbs. These are the useful for the move one place to the other place. The above observation suggests that movement and locomotions cannot be studied separately. Movement and locomotion studied in the same level. It is starting linked between the movements, but all movements are not a locomotions. All movements are not a locomotion. The methods of the locomotion performed in animals vary from the based on their habitat. Based on the demand of the situation, demand of the natural condition, demand of the natural factors, they move from the. However, the locomotion is generally for the searching of the. What is the main advantage for the locomotion? What is the necessity for they move from the one place to the living organism, move one place to the other place? What is the main advantage? For the searching of the food material and also shelter and also mate. Mate means they selected for the suitable partners, and also suitable breeding grounds and also favorable climatic conditions to escape from the, or in some areas unfavorable condition, they can move from the favorable conditions. To also to escape from the enemies and predators. To escape from the enemies and predators. These are the advantages of the locomotion. Advantage of the locomotion. See now, types of locomotion. What is the next one? Types of locomotion. What is the types of the locomotion? What are the cells exhibited different types of movements in our body? The cells of the human body exhibit three types of movement. That is nothing but first one, amoeboid movement, second one, ciliary movement, third one, it is known as a muscular movement. Different types of cells exhibit amoeboid movement, ciliary movement, muscular movement in our body. Muscular movement in our body. Some specialized our body, that is known as the macrophages and leukocytes in the blood exhibit amoeboid movement. What is the cells? Macrophages and leukocytes. Macrophages and leukocytes. Another macrophages are nothing but monocytes. What is monocytes? These monocytes and leukocytes exhibit amoeboid movement. How do you exhibit amoeboid movement? For example, it is indicated in our blood capillary. Whenever these monocytes and leukocytes present in the blood capillary, these are the small in size. These blood capillaries are surrounding of the tissues. But here is looking surrounding of the tissue. Surrounding of the tissue. Whenever these macrophages or monocytes, leukocytes enter into the tissue, the size of the cell is extend. The size of the cell is increases. That is nothing but macrophage. The monocytes only, the leukocytes only, they reaches into the tissue, they engulf the pathogen. They engulf the pathogen, microorganism. In more microorganisms are located, they engulf the pathogen. They engulf the pathogen. This type of movement exhibited in there looks like it. Whenever these monocytes reaches into the tissue, how they will modify it? Like amoeba shape. But it is not a amoeba. Monocytes only modified into the large cell is known as macrophages. What is the necessity for they are modified into the large cells? Engulf the pathogens. Okay, next one. It is affected with the help of pseudopodium formed by the streaming protoplasma as in the amoeba. Okay, cytoskeletal elements like microfilaments also involved in the amoeboid movement. Amoeboid movement. What is the cytoskeleton? See now. Cyto means cytoplasma. Skeletal elements, actin and myosin filaments. Actin and myosin filament. For example, it is one cell. It is indicate other cell. Between the one cell and other cell, the cytoplasma, towards the cytoplasma, what is actin and myosin filaments. Actin and myosin filaments, thick and thin filaments are present. These are present in the cytoplasma of the cell. This related to the skeletal muscles or skeletal elements. Actin and myosin filaments, they exhibit, they also looks like a amoeboid shape. They are also look like a amoeboid shape. They looks like also this micro means the small filaments, actin and myosin filaments. They look like a amoeboid shape. It is one cell, it is the other cell. It is indicated. Next one. Ciliary movement occurs in the internal tubular organs like lined with the ciliated organism. In tubes means respiratory tract. What is respiratory tract consists of the ciliated epithelium. The coordination movement of the cilia and the trachea, they help for the capturing of the removed dust particle and some of the foreign substances along with atmosphere air. It is the ciliated muscles are present in the respiratory tract. The second one, the passage of the ova from the female reproductive tract. It is also ciliary muscles. Where it is? What is the location of the ciliated epithelium? That is the trachea of the respiratory tract, and also ovum. The ovum from the whenever the mature ovum produced from the ovary, the ova egg cell enter to the ovary to the fallopian tube with the help of ciliated. Okay, next one, movement of the our jaw. What is movement of our limbs, jaws, tongues require for the muscular movement of the, it is known as our limbs, skeletal muscles, jaws, again tongues require for the muscular movement, and other movements of the human beings. Majority of the multicellular organisms nothing but locomotion requires perfect coordination of the muscular, skeletal and also neural system. In this chapter we will learn about the types of muscles and the structure, mechanism, and also the contraction of the important steps of the skeletal muscle. Coming to the next one, coming to the muscle. Coming to the muscle. Okay, that is we have a study of the 8th chapter, series of flagellated outgrowth of the cell membrane. The flagellar movement helps in the swimming of the spermatozoa. What is swimming of the spermatozoa, and also maintain the water current of the system of the sponges. Water current system of the sponges. We can indicate any sponge. We can indicate sponge. It is nothing but ostium. What is ostium? These are the indicate ostia. Indicate ostia. It is indicate ectoderm. The next one, it is known as a mesoderm. Mesoderm is the absent. Ectoderm and endoderm present. Ectoderm, endoderm present. This endoderm is consists of the collar cells, are also known as the flagellated cells. What is the cells? Flagellated cells. These cells, flagellum movement of the swimming of the spermatozoa, and also the maintenance of water current in the canal system of the this will the flagellated, the food materials transported into the entire body cell. This flagellated cells are act like a circulatory system. It is act like a heart. Okay, coming to the locomotion of the protozoa like euglena also. This locomotion of protozoa euglena consists of the that is known as the flagella. This flagella also helpful for the locomotion and capturing of the food material. The muscle is the tissue is the originated from the mesodermal origin. The muscle, it is originated from the mesoderma. The muscle is originated from the mesoderma. 40 to 50% of body weight of the only muscles. The 40 to 50 percentage of the 40 to 50 percentage of bodies occupied muscles. They have the specialized properties like excitability. Whenever they receive any signal from the neurons, their excitability. Coming to the next one, after the excitability, contractibility, contraction of the muscles. You can indicate cardiac muscle, always contraction and relaxation, contractibility, and also extensibility. The muscles are easily extensibility, contractibility, extensibility, easily, and also elastability. Elastability means elasticity means they can stretchable. They can easily stretchable into the muscles. These are the properties. Stretchability, that is nothing but elasticity. What is the first one? Contractible, that is excitability. Next one, contractibility, extensibility, easily extend, and also elasticity means easily stretchable. Easily stretchable. The muscles are classified into the different criteria based on the location, based on the nature, regulation activities, based on the location. Three types of muscles: skeletal, visceral, and cardiac muscle. In these muscles, the differentiated between the, we are already discussing at the time of the animal tissues. We already sent into the video of the skeletal muscle, visceral muscle, cardiac muscle. The skeletal muscle and visceral muscle. The visceral muscle are also known as a smooth muscle. The visceral muscle are also known as a smooth muscle. Coming to the next one, cardiac muscle, heart muscles. Coming to the first one. Coming to the first one, skeletal muscle. What is the first one? Skeletal muscle. The skeletal muscle closely associated with the body. The muscle is connected with the bone. They have stripped appearance. Stripped appearance means they have stripped appearance. They have stripped appearance means the skeletal muscle. See the skeletal muscle, the stripped, they always cylindrical shape. Stripped appearance means dark bands and light bands. It is dark band, light band. Dark band, light band. The dark band and light band. Stripped means the striations. Now the striations appears in the dark band, light band, dark band, light band. This appears in the cytoplasma of the skeletal muscle. Under the microscope, you can observe the microscope. It is called as striated appearance as their activities under the control of the nervous system. And these activities are the control of the nervous system. They are known as the voluntary muscles. Why they indicate the voluntary muscle? With the help of skeletal muscle, you can move from the one place to the other place. Coming to they are primarily involved to the change in the body posture. One we have moved from the one place to other place because of skeletal muscle. These are the voluntary action. Voluntary means they can move from the one place to the other place. Coming to the next one, visceral muscles. The visceral muscles are also known as smooth muscles. They are located visceral organs. They are located in the smooth organs. Where it is located smooth organs? Digestive system and also reproductive system and also urinogenital tracts. Urinary tract, they are located. Locomotions are inner wall of the visceral organs of the alimentary canal. What is it? Alimentary canal and also reproductive tract. They do not exhibit striations appearance. They do not exhibit striations appearance. They always spindle shape. They always spindle shape. The striations are absent. The striations are absent. Hence they are called as a smooth or non-striated. The skeletal muscles are also known as a striated muscles. These also indicate non-striated muscles. Non-striated muscles. Their activities are not under the control of the nervous system, but the control of the autonomous nervous system or peripheral nervous system. It is not under control of central nervous system, but it is under the control of the autonomous nervous system. And therefore known as the involuntary muscles. For example, transportation of the food from the one digestive organ to the other digestive organs, and the gambit through the genital tract with the help of smooth muscles. With the help of the smooth muscles. See now, coming to the next one. Coming to the next one. As the name suggests, cardiac muscles are the located only within the heart. Many cardiac muscles are assembled into the branching pattern. You can indicate the diagrammatic representation of the cardiac muscle. It is always indicate branching appearance. It is always indicate branching appearance. These are the branching appearance. The cardiac muscles, the based on the appearance, the cardiac muscles are the striated. These cardiac muscles are the striated. These are the involuntary actions. Do not control on the directly. It is under the control of autonomous and autonomous nervous system, increasing the heartbeat, decreasing the heart rate, everything. Let us examine skeletal muscle under the mechanism and contraction of the muscle. Can indicate each organized skeletal muscle in our body is made up of muscle bundle. We indicate. See, we can indicate the number of muscle bundle. The first one, it is indicate the skeletal, the anatomy of the skeletal muscle observed with the help of microscope. Observed with the help of microscope. Each skeletal muscle. Each skeletal muscle is called as a common collagen connective tissue is called as a shear here. It is indicate muscle bundles. It is indicate muscle bundle. Each muscle bundle connected with the help of one muscle bundle, other muscle bundle connected with the help of the collagen connective tissue. It is known as a fascia. What is it? Fascia. The muscle bundle, the two muscle bundles are contact with the connective. Collagen connective tissue is known as fascia. Okay, next one. Each muscle bundle contains number of muscle fibers. See, number of muscle fibers. Each muscle bundle contains number of muscle fibers. It is indicate muscle bundle are fasciculus. What is it? Fasciculus. Each muscle bundle consists of the, what is this one? It consists of muscle fiber, or also as a muscle cell. Each muscle fiber, it is indicate muscle cell. Okay, each muscle fiber is lined by a plasma membrane. Each muscle fiber is membered by the sarcolemma. Here, each one, it is lined by this one. Each one is lined by that plasma membrane is known as each muscle cell. It is lined by the sarcolemma. Okay, and also it is enclosed by the means. It consists of the cytoplasma of the muscle cell is known as the cytoplasma of the muscle cell is known as a sarcoplasma. For example, it is indicate one muscle cell. It is indicate one muscle cell. The plasma membrane of the muscle cell is known as sarcolemma. The cytoplasma of the muscle cell, it is known as a sarcoplasma. Okay, come to the next one. It is muscles, it is known as a syncytium. What is the meaning of syncytium? Syncytium means the single skeletal muscle cell consists of the more than one nucleus is known as a syncytium. It is consists of the many nucleus. Many nucleus. It is nothing but syncytium. What is syncytium? The nucleus, the single muscle cell consists of the more than one nucleus is known as a syncytium. Next one, as the sarcoplasma contain many nucleus, the endoplasmic reticulum of the muscle cell is known as a sarcoplasmic reticulum. The endoplasmic reticulum of the muscle cell is known as a sarcoplasmic reticulum. Okay, now coming to the next one. Sarcoplasmic reticulum. Next one. Okay, now the reticulum muscle fiber also. What is the function of this one? Calcium ions stored. They are stored for the calcium ions. And also the characteristic feature of the muscle is present in the large number of parallelly present in the filaments are arranged. The large number of filaments are arranged. The large number of filaments are arranged. The large number of filaments are arranged in the muscle is known as a microfilaments. There is nothing but myofibrils. Each myofibrils are indicate dark bands and light bands. It is indicate dark band. It is indicate light band. Dark band means myosin. Light band means that is known as a active. Light band means active filaments. See please indicate a detailed structure of the myofibril, nothing but it is due to the disturbance of the actin and myosin filament. The light band contain actin, okay, the light band contain actin and is called as a isotropic band, whereas the dark band it is known as a anisotropic band. Anisotropic means it consists of myosin. Myosin. Both proteins are arranged in the rod-like structure parallel each other and is called as the longitudinal axis of the myofibrils. The actin filaments are thinner as compared to the myosin filament. Actin filaments are thin, myosin filaments are the thick. Hence, they are commonly called as a thin filament and thick filament. Thin means actin, thick means myosin. Okay, the filament respectively, the center of the eye band, the elastic jet line is known as the elastic biceps. See now, it is indicate jet line. It is indicate jet line. What is the function of the jet line? It is a joining of the two actin filaments. It is a joining of the two actin filaments. It is a joining of the two actin filaments. It is a joining of the two actin filaments. Between the actin filaments, which one is there? Myosin. What is it? Myosin. It is indicate actin. It is indicate actin. It is indicate actin. Two actins are connected. That is nothing but biceps. What is it? Biceps. The red line, it is connected. It is a joining of the two actin filaments. It is joining of the two actin filaments. It is indicate jet line. It is indicate jet line. Okay, now the thin filaments commonly attached to the jet line. The thick filaments of the A band also to the middle of the band. Fibrous band is called as the M line. See now, the middle of the it is known as a thick filament. Thick filament. This one it is middle of the thick filament. It is known as a M line. It is indicate M line. The middle of the thick filament is consist of the M line. What is the M line? A band, I bands are arranged in the order throughout the length of the myofilaments. A band and I bands. This one only actin filaments in I band. Actin and myosin. It is indicate A band. They are arranged. I band, A band, I band, A band. Okay, now coming to the next one. The portion of the between the two successive jet lines is called as a that is nothing but sarcomere. What is it? Sarcomere. Between the two jet lines is known as a sarcomere. What is it? Two sarcomeres. It is the jet line. It is indicated. It is indicate jet line. It is also indicate jet line. Okay, this jet line connected to the actin. The jet line is connected to the actin. Okay, actin. See now, this one only actin filament. Only actin filament. Only actin filament. It is indicate I band. Okay, see, only actin filaments. It is indicate I band. Actin and myosin. Actin and myosin. Actin and myosin. Now this overlapping, it is known as a band. It is known as a band. Now this is only myosin part. See now, only myosin part. Only myosin part. It's zone. Hansen's zone. It is indicate name of the scientist. Hansen zone. Now the middle of the H zone. Now the middle of the H zone is known as a M line. The middle of the H zone is known as a M line. Only myosin and H zone. Only actin, I band. A band means actin as well as myosin. The middle of the H zone is known as the M line. What is it? M line. Okay, now. That is all. At the time of the resting condition, at the time of the resting condition, the thin filaments free ends thick over the central gap of the not overlapping for the H zone. At the time of the resting condition, the actin filaments are only less overlapping of the myosin. Less overlapping of the myosin. Okay, at the time of the resting condition. At the time of the contraction, the actin filaments are more overlappings. At the time of the rest condition, less overlapping. At the time of the contraction, more overlapping at the time of the resting condition. See now, the next one. Before going to the mechanism of muscle contraction, we are discussing about the structure of the contractile proteins. What are the types of proteins involved into the contraction of the muscle contraction? What are the proteins? What are the special proteins important role for their participated into the muscle contraction? Coming to the next one, structure of contractile proteins. First one, actin filaments. It is always exist in the form of two actin filament. There is nothing but fibrous or filamentous actin filaments helically coiled each other. Helically coiled each other. The actin filaments, it is one helically coiled each other. It is one. The second one, helically coiled each other. It is exist in the. It is always two F actin helically coiled. Coming to the next one, each F actin polymer of the G actin filaments. Basically the actin filaments are the G proteins. Basically these are the G actins. G actin means globular protein. In the presence of magnesium ions, the G proteins are again modified into these are exist in the form of monomer. This monomer, they are modified into the polymer. What is it? Polymer. These polymers are nothing but fibrous are F actin. Fibrous are F actin. That is it is exist in the form of monomer. These monomers are modified into the polymer. These polymers are nothing but F actin in the presence of magnesium ions. The next one, two filaments of the another protein is known as a tropomyosin. What is this one? Tropomyosin. See now, this tropomyosin, each is close to the, this is the actin filaments, these are the tropomyosin. This indicate tropomyosin throughout the length. A complex protein troponin is distributed in regular interval of the tropomyosin. See now, the troponin, that is also complex protein. This troponin is consist of the first one is known as a troponin 3. Next one, troponin I. The last one it is indicate troponin C subunit. The troponin protein consists of the how many subunits? 3 subunits. Tropomyosin how many? 2. Okay, now the F actin consists of 2 F actin filaments. These are the proteins involved at the time of the muscle contraction related to the thin filaments. In the resting condition, the subunit of the mass, the active binding site of the myosin on the actin filaments. At the time of the resting condition, you indicate one actin filaments. This one actin filament is consists of the active site. At the time of the resting condition, it is indicate actin. Actin. At the time of the rest condition, at the time of the contraction time, at the time of the contraction time, at the time of the resting condition, troponin T and also troponin I and troponin C. These three complex structures to cover the active site of the actin. To cover the active site, mask the active site of the actin. Whenever at the time of the muscle contraction, more calcium ions are produced into the sarcoplasma of the muscle. These calcium ions bind with troponin C. Whenever the troponin C bind with calcium ions, this entire complex move forward. This at the time active site of the actin is exposed. Whenever the active site of actin is exposed, this easily the myosin head easily bind with actin filaments. It is a formation of the cross-bridge formation. Cross-bridge formation at the resting condition and the subunit protein mask the active site of the myosin head. It is a mask. Next one, each myosin. Each myosin filament is the polymer protein. The basically myosin is the, what is the monomer? That is nothing but meromyosin. What is the monomer? It is the monomer of the myosin. This meromyosin, with the help of magnesium ions, it is modified into the myosin. It is the polymer. It is indicate polymer. It is indicated polymer. The meromyosin modified into the meromyosin. It is basically monomer. Okay, in the presence of many monomeric meromyosins modified into the one thick filament that is known as a myosin. Each meromyosin two parts that is known as a globular head and short arm. Globular head and short arm. See, it is called as a heavy meromyosins and light meromyosins. Now, what is the meaning of heavy meromyosin and light meromyosin? Heavy meromyosin means more number of meromyosins are located in the particular area. That's they indicate heavy weight. Weight is more means more meromyosins. Light meromyosin. Light means the meromyosin number is less. The meromyosin number less means less weight. Light meromyosins. Next one, components and short arm regular distance angle to the polymer myosin filament is known as a cross arm. The globular head, it is consists of the active ATPase enzyme and also site for the binding of the active. What is the active site? See, once here. See, the head of the myosin, and also cross head plus neck. It is known as a head plus neck. It is known as a cross arm. Now this one indicate light meromyosin. Now this one indicate tail. See, the head of the myosin consists of the actin binding site and also second one ATP binding site. ATP binding site, active binding site, and also ATP binding site. Now this one indicate head plus cross arm. This entire part is known as a heavy meromyosin. Now the heavy meromyosin means it is consists of the more number of the meromyosin. That's what it indicated. Meromyosin. Heavy meromyosin. Light meromyosin means is indicate light meromyosin. Light meromyosin means the meromyosin number is less. Meromyosin number is less. That's what indicate meromyosin. The next one. Meromyosin means monomer. The group of meromyosin joined is known as a myosin filaments. Now coming to the next one. Coming to the next indication. Mechanism of the muscle contraction. The mechanism of the muscle contraction. And the mechanism of muscle contraction is explained by the sliding filament theory. Now, what is the meaning of the sliding filament theory? Now, for example, it is one slide. It is indicate other slide. The first slide indicate actin. The second slide indicate myosin. The first slide indicate actin. The second slide indicate myosin. Actin means thin filament. Myosin means thick filament. What is it? Myosin means thin filament. The actin filament slide over the myosin. What is the meaning? Sliding filament theory means it is the actin filament. It is the myosin filament. Actin filament slide over the myosin at the time of the muscle contraction. This muscle contraction is initiated by the central nervous system. It is initiated by the central nervous system. The central nervous system consists of motor neurons and sensory neurons. The motor neurons, they will send message to the central nervous system to the muscle. At the time, these actin filaments are activated. These actin filaments are slide over the myosin. Slide over the myosin. That is nothing but synonym. At the time of the contraction takes place sliding of the thin filament. What is it? Thin filament slide over the myosin. The actin filaments and thin filaments thin filament slide over the myosin. Thin filaments slide over the myosin. That is known as a sliding filament theory. None thick filament, they are not movement now. Only thin filament slide over the myosin. It is a thick filament, it is a thin filament. At the time of the muscle contraction, thin filaments slide over the myosin. Thin filaments slide over the myosin. Coming to the next one. See now, this muscle contraction is initiated by the muscle contraction is initiated by the muscle contraction is initiated by the signals. The muscle contraction is initiated by the central nervous system. This central nervous system, they will send message to the motor. They will send message to the motor neurons. What is the neurons? Motor neurons. What is the neurons? The motor neurons. The motor neuron, they will send, they receive message from the central nervous system. They will send into the muscles now. They will send into the muscles. How they will send into the muscle? It is indicate muscle cell. Or also known as a muscle fiber. It is indicate muscle cell or muscle fiber. See now, the via the motor neurons along the muscle fiber is called as a motor unit. This contact, the contact between the muscles and the contact between the muscles and motor unit and muscle is known as a, this contact is known as a motor unit. What is motor unit? This motor neuron contact with the muscle cell or muscle fiber is known as skeletal muscle is known as a motor unit. The junction between the motor neuron and sarcolemma of the junction between the motor neuron, this junction is known as a, the junction between the, this is sarcolemma, between the motor neuron is known as a neuromuscular. This junction is known as a neuromuscular junction. What is neuromuscular junction? The junction between the motor neuron and the sarcolemma, that is a plasma membrane and the muscle is known as a, that is known as a motor unit. Okay, muscle fiber is called neuromuscular junction. It is also known as a, what is the other name? Motor template. The neural signal reaching this junction, a neurotransmitter which generate the action potential of the sarcolemma. Come to the here. It is indicate synaptic vesicles. These synaptic vesicles are filled. This synaptic node. What is synaptic node are also known as a axon terminal. See, this synaptic terminal or axon terminal is consist of the axon terminal or also known as a synaptic node. It is filled with synaptic vesicles. It is filled with synaptic vesicles. And these synaptic vesicles are filled with neurotransmitters. And this filled with neurotransmitter. It is example for the neurotransmitter acetylcholine. Acetylcholine. Whenever the central nervous system, they will send message to the motor neurons. Here calcium channels are open. This calcium enter into the inside. Whenever the calcium is enter into the inside, this calcium bind with the calcium ions. See, the calcium ions bind with synaptic vesicles. The calcium ions bind with synaptic vesicles. With the help of calcium ions, the synaptic vesicles move into the presynaptic membrane. Whenever they move into the presynaptic membrane, the rupturing of the synaptic vesicles are released into the neurotransmitter. The synaptic vesicles released into the, this space is known as a synaptic cleft. It is indicated synaptic cleft. The outer surface of the circle, that is postsynaptic membrane, consists of the that is known as a acetylcholine receptors. What is acetylcholine receptor? This acetylcholine receptors are bind with acetylcholine. Whenever the acetylcholine receptors are bind with acetylcholine, this receptor located in the postsynaptic membrane. This entire sarcolemma is depolarized. Entire sarcolemma depolarized. Inside the sarcoplasmic reticulum is present. This sarcoplasmic reticulum is open. It releases into the calcium ions. This calcium ion concentration increase in the sarcoplasma. Calcium ion concentration increase in the sarcoplasma. These calcium ions are bind with troponin C. Whenever the calcium ion bind with troponin C, this entire tropomyosin, troponin T, troponin C, the entire complex move forward. The active site of actin is exposed. Whenever the active site of actin is exposed, the myosin head, that is active body site, that is bind to the actin filament. Before the cross bridge formation, before the cross bridge formation, the ATP hydrolysis, ATP is converted into the ADP plus inorganic phosphate plus energy. This energy, it is helpful for the binding of the actin and myosin filament. It is helpful for the cross bridge formation and also helpful for the movement of the rotation of the head. Also see this spread into the muscle fibers is called as a release of the calcium ions. Increase the calcium ions bind. This is by troponin subunit. What is the subunit? Troponin C subunit. What is the troponin C subunit? Actin filament, there may be the remaining of the muscle site, utilizing this energy, and also myosin head also consists of ATP is enzyme. This enzyme is helpful for the ATP hydrolysis. ATP is converted to ATP plus A plus inorganic phosphate plus energy. The myosin head now bind with exposed for the cross bridge formation. Before only ATP hydrolysis. After that hydrolysis process releases to the energy. That energy only utilized for the cross bridge formation. Cross bridge formation. Seen over here, these are the actin filaments. These are the actin filaments. It is indicated myosin filament. This actin filament consists of ATP. Consists of what is ATP. Whenever the cross bridge formation, this one ATP is breakdown, and before only ATP is hydrolysis, they produce the cross bridge myosin head. Formation of cross bridge formation. Whenever the ATP is breakdown, what is the release end product? ADP plus inorganic phosphate. One ATP is breakdown, the second new ATP is added. The new ATP is added. Up to the new ATP is added, again the cross bridge is breakdown. That is known as a relaxation. It is known as relaxation time. The cross bridge is breakdown. Again contraction, again cross bridge formation, again breakdown. ATP is breakdown. Listen to the ADP plus PI. Here the ATP formation, but here the breakdown. It is indicated breakdown of the cross bridge. It is the actin filament, myosin filament. Whenever the actin filament, myosin filament, more slide over. Actin filaments exposed sites are exposed to the myosin head. Binding of the, it is the formation of the cross bridge formation. Whenever the ATP is breakdown, the end product is ADP plus PI. Again, new ATP is added. Many ATPs are present in the muscles now. Many ATPs, many ATPs are present. Sliding filament, this sliding rotation, formation of this energy, ATP hydrolysis time, they produce the energy. That energy utilized for the cross bridge formation and also rotation of the head. Rotation of the head. It is the actin filament. The first one, myosin head, bind here, next one, bind here, next one, bind here, next one, bind here. Means always rotation. This only head rotation. Only head. First bind here, after the move. Actin filaments are move, after the bind, after the move, bind. Like this, they can move. Okay, coming to the next step. See, this is the pull towards to the, whenever they pull toward to the actin filament center of the ear band, this actin filament slide over the myosin because of calcium concentration is increases. These jet lines are attached to the actin filaments, pull towards to the, that is the jet line. It is indicated jet lines. These actin filaments, 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 actin filaments. It is indicate myosin. Is indicated myosin. Is indicated actin filaments. It is indicated actin filaments. Actin filaments. These actin filaments slide over the myosin. Actin filaments slide over the myosin. Inward the causing to the shortening of the sarcomere. This sarcomere between the two jet lines is known as a sarcomere. This sarcomere length also decreases. It is known as a contraction. Contraction means the sarcomere length is decreases. It is clear about the step during the shortening of the muscles. Contraction of the eye bands, connection get reduced. See now, eye bands reduced. Next one, A band remains same. Next one, the myosin releasing ATP goes back to the relaxed state. Okay, now it is an ATP binding to the cross-bridge formation. Again, cross-bridge is breakdown. ATP is again hybridized the myosin head cycle to the beginning of the formation. See here? Here, more it is a relaxation time. It is the relaxation time. So now H zones, I bands, A bands. This H zone is disappear at the time of the contraction. At the time of the contraction, at the time of the maximum contraction, two sarcomeres decreases, Z length also decreases, I band also decreases, A band only remains same. Remaining all are decreases. At the time of the breakage of the nerve, at the time of the breakage of the synapse, at the time of the whenever complete calcium ions decreases, again cross breakdown, the continuous calcium ions are back to the cisternae of the, it is the muscle. At the time of the relaxation time, these calcium ions go back to the sarcoplasmic reticulum. Okay, now this one is called again masking of the actin filaments. The Z lines go original position, that is known as a relaxation. Okay, now this reaction time of the fibers are different from the repeated activation of the muscles, accumulation of the anaerobic respiration. At the time of the anaerobic respiration, the oxygen levels are less. Whenever the oxygen levels are less, the glucose is converted to pyruvate is converted to lactic acid. It is causes to the muscle pains. Okay, now breakdown of the glycogen causing to the muscle contraction. The muscle contraction is nothing but red muscle fibers. Next one, white muscle fibers. Now, red muscle fibers means myoglobin is more. Myoglobin is more. Myoglobin is less. More oxygen. Less oxygen. Okay, now it is the sarcoplasmic reticulums are less. Now the sarcoplasmic reticulum is more. It is participating to the aerobic respiration. It is participating to the anaerobic respiration. See, it is myoglobin present in the oxygen storing pigment. Now hemoglobin means transport of oxygen. Myoglobin means storage of oxygen. Or to the reddish appearance is known as a red fibers. These muscles contain plenty of the mitochondria, utilize large amount of oxygen, ATP production also more, and therefore aerobic respiration. On the other hand, some of the muscles are very less quantity of the myoglobin, that is known as a appearance of the white in color, but these are white fibers, number of mitochondria is also less, and also they participate into the anaerobic respiration. What is the main difference between the red muscle fibers and white muscle fibers? Red muscle fibers and white muscle fibers. Red muscle fibers are less amount of the red muscle fibers are less number of sarcoplasmic reticulum, and white muscle fibers are more number of sarcoplasmic reticulum. Red muscle fibers are aerobic respiration. That is white muscle fibers are participating into the anaerobic respiration. Thank you very much.